Hi, this is your host Alexander with SSRI Side Effects, and today we will be covering my research interpretations from Roberto Cosimo Melcanghi's paper. This includes research on how PSSD and PFS, post-finasteride syndrome, are closely linked with one another. This may even have a carryover to yet another one known as PAWS, post-Accutane withdrawal syndrome. There is suggested involvement of an enzyme converting norepinephrine into epinephrine. For example, PFS can also be caused by other 5-alpha reductase inhibitors like dutasteride. This enzyme converts testosterone into DHT. All the medications I mentioned earlier are able to cause side effects that persist even after discontinuing them. Steroids are a type of hormone. They can be secreted by different glands in the body, even the GI tract, also known as the gut. These are known as corticosteroids, for example, cortisol. However, a type said to play a role in PSSD are called neuroactive steroids. A few functions of neurosteroids include modulating neural plasticity, learning, memory, and behavioral processes. This paragraph here explains the production of steroids from cholesterol and a whole different set of complex pathways. However, my key takeaways are that there are a number of dysregulated enzymes responsible for converting these neurosteroids to subsequent ones and eventually their relevant metabolites. So ultimately, any curative efforts would not be likely to fine-tune every single enzyme perfectly. Rather, there would have to be augmentation of the gut microbiome, possibly through personalized probiotics. A metabolite of progesterone found to be very important is allopregnanolone, and it appears to occur at high levels in the guts of affected rats. It binds to GABA A receptors, which can be found throughout the gut and the brain. This brings us to our next segment, the gut-brain axis. The connection between the gut and the brain is often referred to as the gut-brain axis. It is also called the enteric nervous system, which is actually the lesser-known third branch of the autonomic nervous system. Interestingly, the colon is reported to be able to produce steroid hormones. The composition of the microbiome seems to be important in doing this. More evidence is growing about the importance of healthy and diverse microbiota in the function of our immune systems, as well as brain and neurological function. SSRI antidepressants and possibly SNRIs are able to cause PSSD. The mechanism of sexual dysfunction caused by SSRIs is not exactly known, but a plausible explanation is that the excess of the neurotransmitter serotonin blocks the release of dopamine, where receptors in circuits of the brain facilitate sexual behavior. Various brain scans show that the activity of these circuits are decreased in SSRI users. Paxil, also known as paroxetine, for example, was found to negatively affect male fertility. There are clear imbalances between the levels of neurosteroids where often one region is high and the other low. Decreases in allopregnanolone, specifically in the gut, is linked with inflammation of its lining. There is a notable decrease in Clostridia, Gastroenterophilales, Actinobacteria, and Bifidobacteria species in the animal model. While more is being discovered about the importance of the human microbiome in our health, everyone has a slightly different one, so it is essential to document each other's composition so we can find future possible treatments of diseases associated with it. Our next segment is on the topic of post-finasteride syndrome. Many affected patients reported severe erectile dysfunction. They often showed vascular issues as well as abnormal nerve signals. Genetic polymorphisms were often found in PFS patients in terms of how their bodies make androgen receptors. These are the receptors on which testosterone binds to exert its effects. A mix of underexpression and overexpression were found in this cohort of patients. Additionally, there have been epigenetic mechanisms proposed. Analysis of methylation indeed confirmed it was present on SRD5A2, a gene that produces a type of 5-alpha reductase. It was far more frequently methylated in PFS patients. In other words, this gene was shut off. It makes sense, though, because a medication is in the body which ceases the activity of the enzyme, so negative feedback just tells the body to stop producing it. Once again, gut dysbiosis was found in PFS patients. Overall diversity in bacterial colony counts decreased over many species. Changes in the microbiota can contribute to the development of depressive symptoms and even, presumably, sexual dysfunction. As we can see in this paragraph, all the hormones and levels are really thrown out of balance in the mouse model of finasteride treatment. The gut species in rats found a decrease in firmicutes and an increase in bacteroidetes. The increase is found to be linked with inflammatory bowel disease. It's now known that many neurotransmitters are created in the gut, and after finasteride withdrawal, inflammation occurs, decreasing dopamine while simultaneously increasing serotonin levels. Dopamine acts like a gas pedal, promoting sexual function, where serotonin acts like a brake pedal, preventing this. The figure shows an overlap with PSSD and PFS etiopathogenesis. Both paroxetine and finasteride inhibit PNMT, an enzyme that converts norepinephrine to epinephrine, which I mentioned near the beginning of the video. With numerous similar syndromes like this, we should learn to be vigilant to see if any other types of medications happen to be risky. Once again, I want to stress that more documentation of the species present in each person's gut microbiome would be important to potentially avoid issues and find better alternatives. Do you guys have any research interpretations? Leave a comment down below and we can discuss them. Remember to leave a like, subscribe, and share this video all around. I hope you enjoyed it and learned something about what's going on with the current research. I appreciate your viewership. Thanks for watching.